Welcome, Dr. Sadhana.
मला वाटतं काही गरज नाही आहे मला उभं राहायची.
She doesn't really need any introduction, but I've been given the task to introduce her. So Dr. Sadhana Patwardhan is the director of Nagpur Test Tube Baby Center and a consultant obstetrician and gynecologist there. She is a recipient of Raj Bahadur Narsingh Das Gold Medal for coming first in university in her final MBBS. She is the editor of newsletter of Maharashtra chapter of ISAR, honorary librarian at Maharashtra State Chapter ISAR 2016 to 18, member Foxy Infertility Committee, member Indian Society of Assisted Reproduction, member of ESHRE, and National Faculty for ISAR and CCreate. Welcome, Dr. Sadhana.
Thank you, Chairpersons. Good afternoon, everybody. This session has been really interactive and interesting because Nandita told us how to salvage the ovary, how to give the maximum possible output from the ovaries to the patients. Then Sunita took over us to the journey of stem cells in rejuvenating the ovaries. Then we came down to Matthews talking about the freeze-all strategy and how to give the best possible results in normal responders, poor responders, high responders. Padma Rekha summarized by talking about I-cause. What I am going to do in the next 20 minutes is going to take you into an entirely different journey of genetics of folliculogenesis and premature aging of oocytes. I'm very happy that Deepak is there as a chairperson because wherever I may not have connected well with the topic, he is there to fill in the gaps. Actually, this is more of a job of a geneticist and a biomolecular person, but I chose this topic to have an insight into an entirely different field. So when we talk about genetics of folliculogenesis and premature aging of oocytes, we just need to revise that the carefully orchestrated progression of the oocyte through meiosis has been the subject of several reviews in which essential genes have been identified. A second major transition occurs perinatally when the flattened granulosa cells surround the individual germ cells to form primordial follicles in which the central oocyte is encased by a single layer of flattened granulosa cells and bounded by a basal lamina. The transition from this primordial to primary follicle is a key event in folliculogenesis and thus represents a crucial checkpoint in female reproduction. Now, identifying these pathways, controlling these early events may provide the basis for the fundamental mechanisms that regulate the follicle and potentially lead to new therapeutics. Insight into these signaling processes may provide ways for rescuing the ovarian reserve in risk of women with premature ovarian insufficiency or the ones who are going through chemotherapy. Now, these primordial follicles constitute the full complement of eggs which are available for the subsequent fertilization. Another important point which we need to know is the germ cells which fail to interact with the gonadal somatic cells either by ectopic location or in the absence of a critical oocyte-specific factor do not survive. And the cohorts of primordial follicles are recruited with cyclical periodicity and successful folliculogenesis involved cells and non-cells autonomous genetic hierarchies. So just let us have a look into the different oocyte-specific genes which are responsible for folliculogenesis, which are responsible for fertilization, and which are responsible for embryogenesis. This is again a description of the different genes which I have just put in a tabular format. Now let us see how and why these genes are important and why we actually need to know them. The list of oocyte-specific genes required for normal development of germ cells and surrounding somatic cells, it continues to expand every day. These genes are DASLA, Figla, the Sol LH, the Nobox gene, the YBX2 gene, the CPEB and the GDF genes. These genes, they encode a compilation of transcription factors, mRNA binding proteins and secreted proteins required for normal germ cell development. The Figla gene encodes an oocyte-specific transcription factor. Although the Figla transcripts are first detected at embryonic age 13.5, this protein is not detected until embryonic age 18 using a sensitive gel mobility shift assay. What do these Figla genes do? They coordinate the expression of ZP2 and ZP3 genes that encode the glycoproteins for extracellular zona pellucida. And why are these Figla genes important? Because the Figla null female mice, they lack the zona transcripts and they have the inability to form the primordial follicles. The other genes which are important are the SAW and the SAW2. These are expressed in the male and female germlines as early as 13.5 embryonic age. The females who lack these genes appear to have a normal embryonal gonadogenesis, but they form imperfect primordial follicles which do not progress to primary follicles at all. Nobox is another important gene which is an oocyte-specific homeobox gene. The Nobox transcripts are detected as early as in gonad development at embryonic gestational age 15. This protein is abundant in germ cell cysts, resting oocytes, primordial follicles and growing oocytes. The female mice who lack this Nobox gene do not have a transition from the primordial to the primary follicles. That transition is severely compromised and the oocytes in the Nobox null females, they degenerate by a pathway other than apoptosis, and female but not the male Nobox null mice are sterile. So just to summarize, this Figla, Dazzler, Nobox and all that, just to summarize, what we need to know is Figla is responsible for initial formation of the primordial follicle. The SAW-LH1 completes this process and the Nobox gene is responsible for progression to primary follicle. Now, whether this is a direct linear progression or there are networks of transcription factors, downstream targets, oocyte growths and folliculogenesis factor, this entire gamut, this entire mystery still needs to be solved. Now for folliculogenesis, we need to know that there are intrinsic programs in the oocytes and the surrounding somatic cells. There are intimate communications between the two cell types as I discussed, and functional ablation of this oocyte somatic cell interaction leads to failure of folliculogenesis. The other factors which come into play are the tyrosine kinase receptor, KIT and its ligand KIT-L. GDF9 and bone morphogenic factors are the other factors which mediate the interplay between the oocyte and somatic cells. The oocyte genes which are responsible for fertilization are the ZP1, ZP2, ZP3, and the ones which affect the early embryonic development are the ZAR, NPM, NALP and the DPPA. Now why I am talking all this? This is a reference which I have taken from Deepak and his studies. They have done the studies. They studied a group of 100 cases of patients with idiopathic ovarian failure. So where does this theoretical knowledge of genes and all this come is they have found incidence of one patient out of 86 had an FSH receptor gene deficiency, 3 patients had androgen receptor gene deficiency and 2 patients have had FOXL2 gene deficiency. They could not, however, find inhibin A, Nobox and FMRI mutation in these patients. This is another study which talks about the Nobox variants, the NR5A variants and the Figla mutations which have 2% in the Chinese population but rare in the Asian population. With this, we now move over in another 10 minutes to premature aging of oocytes, because all of us know we have to optimize our protocol. We have to collect minimum number of oocytes to produce, as Matthew said, at least a single euploid embryo and give our patients a pregnancy. But the problem which is now being faced is we are getting more and more of patients with diminished ovarian reserve. The quality of eggs is also not good. So just let us get an insight into what is premature aging of oocyte and are there any rational methods to correct this in the lab. Now the oocytes which are arrested at the M2 stage are normally fertilized soon after ovulation, and the window for fertilization in human is around less than 24 hours. Now what happens if fertilization is not occurring within that time? The unfertilized oocytes, they are remaining in the oviduct or in the culture, they undergo a time dependent deterioration in quality, a process which is called oocyte aging. Now oocyte aging, however, occurs in ovaries of females who show a progressive decline in fecundity and also have a poor developmental potential of aged oocytes. Now when we discuss the mechanism underlying this oocyte aging and possibilities to reverse oocyte aging, which would have a significant impact on the assisted reproductive technology, we just need to see three important headings. Like what are the abnormal morphological and cellular changes in these aged oocytes? What are the mechanisms underlying the oocyte aging? And are there any practical methods to reverse and control these oocyte aging? Now this is just a table which summarizes the changes in the morphology and appearance of the fresh oocytes and the aged oocytes. The zona pellucida is a granulofibrillar structure in fresh oocytes. It's a cobblestone appearance in aged oocytes. The perivitelline space is small. It is large in the aged oocytes. The spindles, they display a vertical orientation to the oolemma. The spindles, they are elongated and smaller with few microtubules in the aged oocytes. The chromosomes are intact, arranged symmetrically. They display a PCS and they are scattered throughout the degenerating spindle. The mitochondria in the fresh oocytes are intact, but the mitochondrial matrix shows remarkable swelling in the aged oocytes. Now why we need to know? Because these functional changes associated with oocyte aging, they have an implication in the lab. When your embryologist is telling you that the fertilization has not gone very well, are we dealing with oocytes which have prematurely aged? We never know. Because these are the oocytes which have decreased fertilization rate, they have polyspermy, they have parthenogenesis, chromosomal anomalies, apoptosis, epigenetic changes, and an abnormal or a retarded development of the embryos or fetuses. So now a variety of factors play a role in oocyte aging, including the environmental conditions, oocyte interactions with the cumulus cells, and various chemical components. New and encouraging data have shown that a variety of chemicals can reversibly regulate this oocyte aging, and managing oocyte aging by supplying such chemicals to the culture media may particularly be critical to improve the modern technology. The different chemicals which I came across when I studied this particular topic is L-cysteine, which decreases the oocyte development to the blastocyst stage. B-mercaptoethanol, it decreases the development to the blastocyst stage. Vanadate, it accelerates the oocyte aging. There have been results that caffeine inhibits oocyte aging. Nitric oxide delays the oocyte aging. DTT, that is diethiotriol, it increases the fertilization rate and number of embryo cells, and porcine use of TSA has shown reduced oocyte fragmentation and increased developmental capacity. Now, not going much into the theories of all this, and there are subtle other physicochemical factors like temperature, presence of cumulus cells, and the reactive oxygen species which also affect the aging of oocyte. Just to give a brief introduction to this is whether oocyte aging is a prelude to apoptosis induced by the calcium elevation, because a lot of work is being done now as to the application of the calcium ionophore and oocyte activation. So just a brief slide about this. In mammals, at fertilization, the sperm activates the oocyte development by initiating a prolonged series of elevations in the intracellular calcium generated by the increased production of the IP3. Now what happens actually in an aged oocyte, the frequency of this calcium elevation is higher, but the amplitude of oscillations is smaller. So there is the dysfunction of the endoplasmic reticulum of this calcium pump, and this fertilizing calcium signal is inadvertently now converted to an apoptosis inducing signal. So this is what exactly is occurring in a aged oocyte. This I am talking because a lot of people are doing calcium activation, calcium ionophore activation for the oocytes. Now there are certain abnormal epigenetic changes which are associated with oocyte aging. As far as temperature is concerned, we know that there are negative effects of temperature changes on the mouse oocyte aging which have been shown. The long-term effects of oocyte vitrification on oocyte aging, there is yet no long-term data available. We also know that cumulus cells are critical for oocyte maturation, ovulation and fertilization, and they do protect the oocytes from the damage from the ROS. So now are we anyway getting closer to how to reverse the oocyte aging? There are certain other factors like reactive oxygen species, superoxides and HOCl. They also augment the oocyte aging. This is another pathway, will not go much into the details about the MPF and the pre-MPF pathway, where what we need to know is oocyte aging, to summarize, can be reversed by controlling the activity of this MPF. Investigators have found that both MPF and MAPK are important for maintaining the oocytes arrested at M2 stage. Vanadate and caffeine can modify oocyte aging differently by controlling this MPF activity. Vanadate accelerates oocyte aging, caffeine delays oocyte aging, nitric oxide prevents oocyte aging. I have presented an entire gamut of chemicals, but the human oocytes, there are a lot of ethical concerns, limited availability. So there is a big doubt whether this can be practically made applicable in our lab, and practicability of this application is not yet proven. So then what? The path-breaking research now which is being done in most of the countries is transferring cytoplasm from the fresh oocyte containing fresh mitochondria and other organelles in order to replenish the essential cellular components, the BCL2 and the mRNA involved in regulation of the apoptosis program, addition of the fresh mitochondria into the aged oocytes in patients who have had mitochondrial dysfunction. But still, to conclude, a number of chemicals have been employed to delay oocyte aging and provide promising new possibilities for intervention in this aging process. For the ART procedure to have a successful outcome, we need optimal production of normal and healthy babies, cytoplasm transfer, mitochondrial transfer, and analysis of oocyte-specific genes by gene ablation and transgenesis is the future. Maybe few years from now, we'll have more of Deepak and his colleagues talking here about the transgenesis and gene therapy in ART, rather than we just discussing protocols. Investigating these processes will not only enlighten our knowledge of the normal biology, but will also provide insights into disorders that affect human reproduction. And I thank you all for a patient hearing.
Thank you so much, Dr. Sadhana. That was very, very informative, because as a clinician, it gives us an idea what to tell a patient, especially in those unexplained infertility cases where you get oocytes, you have a decent number, 8 to 10, but even with ICSI, you have failed fertilization. So one of the reasons could be a bad oocyte. We just say bad oocyte, but now we could talk of genetic factors ki ye ho sakta hai. Second thing what we see is the younger Indian population getting premature ovarian failure. Maybe some factor applicable there. What do you think, Dr. Deepak?
Yes, there have been a bunch of studies where they suggested that increased mobile phone usage and changes in circadian rhythms is associated with poor oocytes, because these girls these days tend to sleep lesser, stay awake late night, and that somehow affects the oocyte and the ovarian circadian rhythm. And that's one of the reasons why so many such changes we keep seeing these days. So there was a study published from India a couple of years ago where they've shown that in girls with PCOS, there is increased expression of genes associated with circadian rhythm, and that directly correlated with the late night mobile phone usage. So we must counsel our patients in this case to have the circadian rhythm set, have the nice 8 to 9 hours sleep at the right time, not just at any time. So that would be one good take-home message.
Thank you so much. Wonderful speakers, wonderful audience. Any questions from anyone?